So today and tomorrow we're going to be talking about transcription and translation. So yesterday we learned about DNA replication, so how cells replicate their DNA, and today we're going to start learning about how cells transcribe that DNA and then translate the message into a protein. So we're going to see how the information that's stored in DNA is actually used. Now there were a couple of simulations to play with, but these are Flash animations, so you may not be able to get them to work because Flash is no longer being supported, so I'm not sure what the alternatives are. But if you have the ability on your browser to activate Flash still, you might want to check out these simulations. So if you just Google nobelprize.org DNA double helix game, it should still be there, but you just may not be able to play it unless you can activate Flash Player. And same thing with translation. If you Googled CPUP protein synthesis simulation, it's there, but again, you might not be able to play it. So that's one of the problems. All these animations were made using Adobe Flash Player, and Flash Player is no more. It's no longer being supported. Some browsers, you can't use it at all anymore. So, finding alternatives will take a little bit of time. So, as I said, we know how the information is stored in DNA. It is stored in the nucleotides. Remember, we have just 4 different nucleotides: adenine, thymine, guanine, and cytosine. Adenine base pairs with thymine, and guanine base pairs with cytosine. So A's with T's and G's with C's. There are stretches or regions of DNA which constitute a gene. And so we're going to learn today, or start learning today, how the information of that gene is read in order to produce a protein product. And it's the proteins that give organisms their characteristics. And when we get into genetics, we'll learn more about genotype and phenotype. This example here, Gregor Mendel was a monk who is sort of considered the father of genetics, even though he didn't know anything about the molecular aspects of what was going on, but he made many observations. And he worked with pea plants. Because pea plants were easy to grow, they made a lot of seeds, and they had some observable characteristics. And some of his pea plants were dwarfs. And they were dwarfs because they did not have a functional copy of a gene for a protein called gibberellin. And gibberellin stimulates growth and elongation of stems. And so some of his pea plants, that gene was not functional. And so they were dwarf. So originally, they thought that one gene coded for one enzyme. So one gene, one enzyme hypothesis. All enzymes are proteins, but not all proteins are enzymes. And so many other proteins are encoded by the genes. And so they tweaked the hypothesis to one gene, one protein. And that protein may or may not be an enzyme. But then later research demonstrated that many proteins were composed of multiple subunits, right? Multiple polypeptides. And so then they came up with one gene, one polypeptide as their hypothesis. So genes provide all the instructions for making specific proteins, and it's all those proteins that enable an organism to exist. And the bridge between DNA and protein synthesis is RNA. So DNA will be transcribed into RNA. Later, the RNA will be translated into protein. RNA is a molecule chemically similar to DNA, but instead of deoxyribose as the sugar, it's just ribose. And instead of thymine, RNA uses uracil. And uracil is chemically similar to thymine. And I think I mentioned last time that uracil is not as chemically stable as thymine is. So RNAs are not very stable. They do not last very long. And that's something the cells want to happen. They do not want the messenger RNAs to last a long time. Because if that messenger RNA is there in the cytoplasm and is constantly being translated to make proteins, maybe the cell does not need those proteins. And so the cell would be wasting energy by translating them. And so messenger RNAs by design do not last very long inside the cell. That's a mechanism of regulation of protein synthesis. Right? So messenger RNAs have to continually be made if the cell wants to continually make those proteins or polypeptides. And RNA molecules are single-stranded most of the time. There are some viruses whose genome is double-stranded RNA, but again, that's one of those exceptions, not the rule. So in DNA or RNA, there are 4 nucleotides. And those 4 nucleotides are like the letters of an alphabet used to communicate information. And the specific sequence of hundreds or thousands of nucleotides in each gene will carry the information for the primary structure of a protein. Remember, proteins have primary, secondary, tertiary, and quaternary levels of structure. The primary structure is just the sequence of amino acids. And so the nucleotides will tell you what the sequence of amino acids in that protein will be. And there are 20 possible amino acids, and we'll see these shortly. To get from DNA into RNA and then to protein, there are those stages of transcription and translation. And so we'll talk a little bit about both today, and then we'll start getting into some more details today and then tomorrow about the 2 processes. But transcription and translation are the 2 major steps. DNA is transcribed into RNA. RNA is translated into protein. During transcription, only one of the DNA strands will be used as a template for the synthesis of complementary RNA. So can anybody tell me what is the enzyme that is going to synthesize the RNA? What's that enzyme called? The enzyme that makes RNA, what's that called? What's the enzyme? Yeah, RNA polymerase, right? So it is very similar to DNA polymerase. And so I'll give you 3 guesses. In what direction is the messenger RNA synthesized? In what direction?
Is it in the 3' direction?
In what direction?
The 3' direction?
So give me the whole phrase. It starts and goes to?
5' to 3'?
Right, so synthesis is in the 5' to 3' direction, just like with DNA synthesis. The RNA polymerase can only synthesize in the 5' to 3' direction. So that means the DNA strand that's used as a template runs in the 3' to 5' direction, right? In the opposite direction. And that way we get synthesis of a 5' to 3' messenger RNA. And the messenger RNA is what we call the type of RNA produced by transcription. There are many other different kinds of RNAs, and we'll talk about some of those in the remaining lectures. Today we'll also talk about transfer RNA and ribosomal RNA. But there are many other RNAs out there, and we'll mention a few of them later on. So during translation, the information contained in the messenger RNA is going to be translated. So the information is going to be read in order to determine the sequence of amino acids in that polypeptide. And the little machines inside the cells that do this are called ribosomes. So the ribosomes are the little protein synthesis machines. They read the messenger RNA and translate it into a polypeptide by joining a bunch of amino acids together. So when it comes to transcription and translation between eukaryotes and prokaryotes, they're basically the same. There are some minor differences, like how we mentioned with DNA replication. 3 main bacterial enzymes, DNA polymerase 1, 2, and 3. Eukaryotes have many different kinds of polymerases, but the general process is the same. So there are some fine details that are different. Now, an important difference, a very important difference, is we know that prokaryotes do not have a nucleus. So the bacterial chromosome, or the prokaryotic chromosome, is in the cytoplasm. That's the same place where the ribosomes are. So what that means is that in bacteria, or in prokaryotes in general, as the messenger RNA is being transcribed, it can simultaneously also be translated. So as the prokaryotic cell is transcribing the DNA into RNA, the end of the RNA that's already been synthesized can hook up with a ribosome and start being translated even before the messenger RNA is complete. In eukaryotes, that doesn't happen because transcription occurs in the nucleus, because that's where the DNA is. And so the messenger RNA is synthesized in the nucleus and is then transported to the cytoplasm, where it hooks up with the ribosome. So there's a delay when it comes to the synthesis of a protein in eukaryotes versus prokaryotes. We're also going to see that eukaryotes have to do a lot of processing to their messenger RNA before it's actually ready to be translated. Prokaryotes don't do that. They don't need to process their messenger RNA in order to translate it, but eukaryotes have to. So the initial RNA transcript in a eukaryote is called the primary transcript. It's going to be modified in several ways before it becomes the final messenger RNA transcript that will eventually be translated by a ribosome. So eukaryotes are more complicated, as we're going to see. So which cell type can make a protein faster, a prokaryote or a eukaryote, and why? So I just told you, but you tell me. I'll see if you guys are paying attention.
A prokaryote, because transcription and translation happen at the same time.
Yep, they happen simultaneously in the cytoplasm. But for eukaryotes, that nuclear membrane separates transcription from translation. So that creates a delay, which is okay for most eukaryotes because most eukaryotic cells belong to a multicellular organism. And so the eukaryotic cells, they have a lot of friends, right, to help them out. But prokaryotes are single-celled individuals. They need to be able to respond more quickly to a changing environment because they don't have anyone else to rely on but themselves. In a eukaryotic organism, the internal environment tends to be a lot more stable, and so cells don't necessarily have to react quite as quickly. So DNA is transcribed into RNA, is translated into protein. The flow of this information in a cell is called the central dogma of biology. So if anyone ever asks you in the future, what's the central dogma of biology? You tell them it's the flow of information in a cell. And the information flows from DNA to RNA to protein. And for many years, that was it, a one-way flow of information. But not all that long ago, we discovered we could go backwards somewhere. Any idea where and how? How has the flow of information been modified? Where can we go backwards? We can go from RNA back to DNA. Anybody know how? I actually mentioned it last lecture. I didn't talk about it, I just mentioned the words. So if the forward direction from DNA to RNA is called transcription, what would you call it to go from RNA back to DNA?
Reverse transcription.
Right, reverse transcription. So can you think of an enzyme that would allow for reverse transcription to occur? I'll give you a hint. It starts with reverse. And it ends with ace.
Reverse polymerase.
You see me writing, don't you? Reverse transcriptase, because it reverse transcribes RNA back into DNA. And that's a very important enzyme. Originally discovered, I think, in viruses, because several types of viruses have reverse transcriptase. Once scientists discovered that enzyme, they said, hey, we can use this. And we can use this to take RNAs and convert them back into DNA. Because DNA is what we clone into cells, like bacterial cells. So now we found an enzyme that allows us to make a gene. And we use this enzyme a lot now. So When it comes to all the nucleotides and the genetic code, we know we have 4 nucleotides, right? ATGC in the DNA, ATGC. So if each nucleotide only coded for one amino acid, A for one amino acid, T for a different one, G for a third one, and C for a fourth one, that's only 4 amino acids. And we know we have 20, so that's not enough. So what if we took our 4 nucleotides and we put them into pairs? How many combinations do you get when you take 4 nucleotides and put them into pairs? Right, so we have 4 nucleotides and we want to pair them. How many combinations do we get?
16.
16. Well, that's not enough because we have 20 amino acids. And we haven't even mentioned start and stop codons yet, so that's no good. But what if we take these 4 nucleotides and put them into groups of 3? Then how many possible combinations do we get?
64.
64, which is way more than we need, right? We only have 20 amino acids, and then we need at least 1 for start and stop, right? So 64 is kind of overkill, but that's the minimum number, right, that's required in order to at least code for those 20 amino acids along with start and stop codons. So if we take our 4 nucleotides and we put them into groups of 3, we get 64 different combinations, way more than is needed. But when you have just 4 nucleotides to work with, groups of 3 is the minimum number needed to get at least 20 unique combinations, plus a few extra for start and stop. So the genetic code is based on 3 nucleotides. Every 3 nucleotides is a codon, also known as a triplet codon. So that's how the information is stored. So remember, DNA was this very simple molecule, a polymer of just 4 subunits, A, T, G, and C. Now they discovered that, well, if we arrange those 4 nucleotides into groups of 3, that's how the cell interprets that information in order to add amino acids to form a polypeptide. So it's kind of elegant, but simple. So we said that only 1 strand of the DNA is used as a template strand to synthesize the messenger RNA transcript. The base pairing rules are still the same, except there is no T in RNA. It's replaced with U. So if we go back to my sequence from last class, and I ask you now for the complementary messenger RNA strand, what would that be?
CU AA U G U.
Right? So wherever you would normally want to put a T, it's now a U. But otherwise, everything else is the same. It's just there's no T in the RNA. It's replaced with a U. But you still have C's, you still have G's, you still have A's. You just have no T's. They're all U's. So is that clear? Is there anything familiar about the sequence here Gattaca?
It's the name of a movie starring Ethan Hawke and Uma Thurman.
You're right. It's the name of a movie. I think it was in the 90s. Yeah, Ethan Hawke and Uma Thurman. Gattaca. And the movie was about genetically modified humans. And so Gattaca was genetic code. How many people do you think in the audience understood that? Do you think very many people in the audience knew what that meant? I don't think so. So during translation, the codons are read in the 5' to 3' direction, along with the messenger RNA. Each codon specifies one of the 20 amino acids, which will be incorporated into that polypeptide. So because the codons are base triplets, right, there are 3 nucleotides. We also just call nucleotides bases, and they're groups of 3. The number of nucleotides making up a genetic message must be 3 times the number of amino acids making up the protein product, right? Because one amino acid requires 3 nucleotides in the codon. So we need 3 times as many nucleotides as there are amino acids. So if you had 300 nucleotides in your piece of DNA, you could code for a polypeptide that's 100 amino acids long, right? Because every 3 bases, every 3 nucleotides codes for 1 amino acid. So if you've got 300 nucleotides, that means 100 amino acids. The average human protein length is about 485 amino acids. So let's make the math simple and round that up to 500 amino acids. How many nucleotides are required to code for that protein? 1,500, right? 3 times 500. So you can see that chromosomes, eukaryotic chromosomes, are incredibly long, right? They are made up of many, many, many nucleotides because humans, on average, have around 30,000, genes approximately. It's just a rough number, not the exact number, but approximately 30,000 genes. So that's a lot of nucleotides, right? So the task of matching each codon to its amino acid began in the early 1960s. So again, I know that's a long time for you guys, not that long for me. But in the 1960s is when they started to try and figure out the genetic code. Marshall Nirenberg was one of the first ones to do this, and he used a codon of UUU. So he had a piece of RNA that was all just UUUUUUUU, all U's. And that ended up coding for 1 amino acid, phenylalanine. So he did this. We created artificial messenger RNA molecules. In this case, one that was made of just uracil. Put it in a test tube with all the amino acids, ribosomes, and everything else needed for protein synthesis. And phenylalanine was the only amino acid produced. Therefore, he determined UUU coded for phenylalanine. And so then they started to do this with all the other different amino acids, creating synthetic RNAs with a specific codon sequence to try to determine what codon matched each amino acid. And they eventually came up with a genetic code, which we see here. So this is our genetic code chart. Shows all the different codons, all 64 of them. Now, 61 of them do code for amino acids. And then we have 3 stop codons and one start codon, AUG. AUG also codes for the amino acid methionine. So one start and three stops. Yeah, question, Zoe?
I thought it was only 20 amino acids.
Well, there are. And if you count, there's only 20 amino acids in the chart, right?
Yeah, but you said 61 of them code for amino acids.
I said what now?
You said 61 of them code for amino acids.
Right, but some of them are duplicates for the same amino acid.
Oh, okay, thanks.
Right. So, but the three stop codons are UAA, UAG, and UGA. That means stop. Only one start codon. So basically, all polypeptides start with methionine, right? Because AUG is start. Now, I have to get to Zoe's question. For example, the amino acid leucine, LEU. How many codons code for leucine? LEU, how many codons are there for leucine?
2.
Really? Sure, Chris?
6.
Right, we got 6.
6.
All those codons just
Oh,
code for the same amino acid, right?
Right.
What about serine, SER? How many codons code for that?
Oh, 6 also.
Yeah, also 6. We got 4 here and then 2 here. So for most of the amino acids, there is more than one codon. So that makes the code redundant. That also provides a little bit of cushion against mutations, right? And for example, leucine, as long as your codon starts with a C and a U, you get leucine. It doesn't matter what the last base is. It could be CUU, CUC, CUA, or CUG. You still get leucine. So if there were a mutation in a codon, it's still very possible that mutated codon still codes for the same amino acid. So there's a little protection, a little cushion against some mutations here. But there's just 20 amino acids, but for almost all of them, except methionine, there's more than one codon. In some cases, there's up to 6 codons for a particular amino acid. So the code is redundant, right? A redundant code. So if I ask you, if I give you these 3 codons, what is the amino acid sequence for these 3 codons? ACG, GAC, and UAC. What are the amino acids? What's ACG? So how do you use the chart? Well, the first base, you look along the left-hand side, and the second base, you go across the top, and then the third base, you triangulate and come down. So A, here's A, CG, here's C, so A, C, where's my ACG, and that codes for THR, which is threonine. You don't need to know what the amino acids are. It would be nice if you did, especially if you're going to take more biology courses, but you don't have to know them for this class, but it'd just be nice if you did. GAC, what does that code for?
ASP.
ASP, right? Aspartic acid. And UAC? I find U, A, and and come down. UAC is tyrosine. So on the exam, you'll be given the genetic code chart. If I give you a sequence of RNA, you need to be able to give me the amino acid sequence that goes along with it, and just the 3-letter code that you see in the chart. Don't need to know what the amino acids are, but just be able to use the chart to give me the amino acid sequence. So can we all do that? All good? Joie, is that good?
Can you please go over it again one more time?
So for the GAC, for example, right, your first letter, the first base, right, the first nucleotide, you find here on the left-hand side. Here's G. A, you go across the top. Here's A. And then you come down and find GAC, and that codes for aspartic acid, ASP. So first letter, look on the left. Second letter, go across the top, and then triangulate and come down and find the third one in the table. So is that clear?
Yes, thank you.
Mm-hmm. So, as I said, the code is redundant. It's not ambiguous, right? There are specific codons for the amino acids, but there is some variation in those codons. So like I said, as long as you got the first 2 letters, then the third one can change and you still code for the same amino acid in many cases. Not every case, but in many cases. So, is that clear? The code is redundant. For most amino acids, there's more than 1 codon. In some cases, there's a maximum of 6 different codons for the same amino acid. And that provides a little bit of cushion, a little bit of protection against mutations, because you can have a mutation and the codon still codes for the same amino acid. Now, in order to read all those nucleotides in the correct groups of 3, there needs to be a starting point. So that establishes what's called the reading frame. Because when you look at a sequence of DNA, it's just one continuous sequence of A's, T's, G's, and C's, right? How does the cell, or how do the RNA polymerase enzymes and the ribosomes know where to start doing something and where to stop doing something? Right? Because that's important. If you start in the wrong place, all your codons are going to be read incorrectly. So there are specific signals which tell the RNA polymerase and the ribosome where to start doing something. So here we see all the 20 amino acids and their 3-letter codes. Like I said, you don't need to memorize these. It would be nice if you knew what they were, especially if you're going to go on to take more biology courses, or if you're a bio major, then you definitely need to know your amino acids. Now, if anyone has taken or is going to take nutrition, you'll hear about essential and non-essential amino acids. Essential amino acids are those that we cannot synthesize. We do not have the genes to make histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. We cannot synthesize those, so we have to eat them. That's why they're considered essential, because we need to get them in the diet. If we don't, we're going to be in trouble. because many of our proteins are not going to be made because we're missing these essential amino acids. The non-essential amino acids, we have the genes for those, so we can make those. What's essential and non-essential varies from species to species. It's not exactly the same. But this is what it is for humans. So just sort of FYI, the 20 amino acids that are found in proteins are called proteinogenic amino acids because they're used to make proteins. There are two other proteinogenic amino acids, selenocysteine and pyrrolysine, but these are not, part of the genetic code. They tend to get incorporated at a stop codon if there are some other signals present. So even though they are part of proteins, they're not encoded by the genetic code. But if anyone asks you, how many amino acids are there? Yeah, there are 20 that are found in proteins, but there's actually many more than just 20 amino acids. There are many other naturally occurring and synthetic amino acids, which are non-proteinogenic, so they're not found in proteins. These are reactants for other reactions, or they're intermediates in a metabolic pathway. So the amino acid has to have the chemical structure, has to have an amino group and a carboxyl group, right? And that makes it an amino acid. So there's many, many amino acids. Only 20 of them are actually used to make proteins. So, that could be a trick question, right? How many amino acids are there? We got to be more specific, right? There's only 20 that are used to make proteins, but there are at least 1,000 others which are used for different chemical reactions, or some scientists have created them synthetically. So just a fun fact kind of thing. So the code must have evolved very early in the history of life. And the reason why is the genetic code is basically a universal code. Now, there are some minor differences in certain organisms, but basically the genetic code is the genetic code. And because it's universal, you can take a gene from a completely unrelated species and stick it into a different species, and it still functions. Here we see an example of the firefly gene, which codes for an enzyme, luciferase, which makes the fireflies glow. You can take that gene, stick it into a plant, in this case, a tobacco plant, which is now glowing, right? Because it has that gene. And so we can make transgenic organisms. A transgenic organism is an organism that has received a foreign gene. So we do this with crops, genetically modified crops, GMOs. They have genes that were cloned into them, for different purposes. In some cases, they clone a gene which produces a toxic protein to insects. And so when insects try to eat that plant, they die because the plants are now making this toxic protein, which is only really toxic to insects, not to humans. Or they can genetically modify foods to enrich them with extra nutrients, right, that they wouldn't normally have, but they've been genetically modified to produce these extra nutrients. So we can clone genes into different organisms. You would go to prison for the rest of your life if you did that, because that would be considered unethical if you made a person glow in the dark. But they have done this with fish. Yeah, right. Not if you did it to yourself, but there are fish called glowfish, which have been genetically modified to produce proteins that fluoresce. With the black lights you can put on tanks to help things to glow. So there are genetically modified organisms with these genes. They don't really glow, but if you shine light on them with the right wavelength, they fluoresce. And so they look like they're glowing. Many of those organisms are banned in other countries, but you can buy them here in the US. Although just FYI, the glowfish are not very hardy, right? That extra protein that they're making, which does nothing except give them this color, is not exactly the most beneficial gene to them. So they don't live as long as plain fish do, but you can buy them. And there were claims that this was done to a rabbit and some other animals. But later on in lab, no, people don't eat them. They're just ornamental, right, to look at. But later on, I think next week, we'll talk about a lab in which we would have made glowing bacteria. So if we were meeting face to face, we actually would have inserted a gene into E. coli to make them glow. So, but we'll see pictures of that. But it would have been fun if we could actually do it. So, but the key point here is that, yep, because of the universality of the genetic code, you can move genes around, right, from different organisms. Last time we talked about Griffith's experiment with transformation, where bacteria can absorb pieces of DNA from the environment. And those pieces of DNA can come from unrelated species, not just other bacteria. So we do clone genes into bacteria. An example, insulin. Who needs insulin?
Diabetics.
Right, so we all need insulin, but diabetics either do not make enough or they don't make any at all. And so they have to inject insulin. Where does that come from? How do they get the stuff they inject? Are you going to volunteer to donate some human insulin? I don't think so. So they used to get it from the pancreas of slaughtered animals, extract it and purify it. But that was not human insulin. So that was not the best kind of insulin to use. So how do you get human insulin? Are they going to stick a large needle into your abdomen, all the way to your pancreas, and suck out some insulin? No, they don't do that either. So what scientists eventually did is they found the gene for human insulin, and they cloned that gene into bacteria. And so the bacteria were little factories to synthesize human insulin. And we just purified it from the bacterial culture. And that's what diabetics use today, is a version of human insulin from cloned bacteria. So we modify lots of bacteria to make things for us that way. And in lab, we'll talk about how to do that. Now, when it comes to some exceptions or some variations, this doesn't need to be expensive. Well, that's going to be a whole big discussion about big pharma and the profiteering they like to do. So the answer is no. None of these drugs, or most of these drugs, do not need to be as expensive as they are. It's just that the pharmaceutical companies want to make huge profits. But we can get into that later. So some exceptions to the code can be found in organisms like paramecia, which are eukaryotic protists. When it comes to certain mitochondria and chloroplasts, they also have some variations, because remember, mitochondria and chloroplasts do have their own chromosome, and they have their own ribosomes, and they do some of their own translating. And so there are some differences there. But still, the majority of the code is the same for every organism. And that suggests that it evolved a very long time ago. It also suggests we all came from a common, universal ancestor. So, common, universal ancestors. So let's talk about the actual synthesis and processing of the RNA. And we're going to be focusing on eukaryotes, right? Because prokaryotes don't have to process their messenger RNA. It's basically good to go. It's ready to be translated as soon as it is transcribed. That's why transcription and translation occur simultaneously in prokaryotic cells. But eukaryotes have to do a lot to the messenger RNAs. So we know that RNA polymerase is the enzyme which synthesizes RNA. It uses only one of the DNA strands as a template to synthesize. We've already said RNA polymerase synthesizes that messenger RNA in the 5' to 3' direction. There are sequences in the genes that tell the RNA polymerase where to start transcribing and where to stop transcribing. So something that students always get mixed up, because the one thing they always remember is start and stop codons. That's just for translation, for protein synthesis. When it comes to transcription, there are start and stop sequences. So not codons, but sequences. The start is called the promoter. So there are sequences. All organisms have promoter sequences. This is where the RNA polymerase binds, so it can start transcribing. When the RNA polymerase reaches a terminator sequence, that tells the RNA polymerase to stop transcribing. Get off. Right? The messenger RNA is done. Bacteria just have one type of RNA polymerase, and it synthesizes all the different kinds of RNAs. But eukaryotes, more complicated, we have 3 types of RNA polymerases, 1, 2, and 3, and they're found in the nucleus. So RNA polymerase 2 is the one that's used to synthesize the messenger RNA. RNA polymerase 1 is used for ribosomal RNA synthesis, because ribosomes are made with a piece of RNA, ribosomal RNA. And RNA polymerase 3 is used mostly for ribosomal RNA and transfer RNA synthesis. So the tRNAs. So these are the 3 main types of RNA. mRNA, which is messenger RNA, rRNA, which is ribosomal RNA, and tRNA, which is transfer RNA. And the tRNAs are the ones that are carrying the amino acids to the ribosome in order to build that polypeptide. So here we see a gene. It's got a promoter sequence. It has a terminator sequence. So the RNA polymerase will hop onto the promoter and start to transcribe. When it reaches the terminator sequence, it falls off, right? Because the gene has been transcribed into RNA, and it's done. Now, in reality, it's a lot more complicated than this. RNA polymerase normally can't always just hop on and start to transcribe. There are all kinds of transcription factors which enhance transcription or inhibit transcription. So it does get complicated when it comes to the mechanism of regulation. But the main idea is a promoter sequence is where the polymerase hops on and starts transcribing. When it reaches the terminator sequence, it falls off. It stops transcribing. And so then we have our messenger RNA transcript. It says completed here, but it's just calling it a completed RNA transcript. not a messenger RNA, because a lot of processing has to occur to this before it becomes the final functional messenger RNA transcript. But there is a promoter sequence and a terminator sequence. That tells the RNA polymerase enzyme where to hop on and start transcribing and where to hop off and stop transcribing. So again, it's just some more detail about the promoter sequence. It's just where the RNA polymerase hops on and starts to transcribe. When it comes to prokaryotes, again, it works a little bit differently. Prokaryotes also have enhancers called sigma factors, which you don't need to know, but it's not like just because there is a promoter there that RNA polymerase will just automatically hop on and start transcribing. There's actually a lot of regulation that's involved in transcription, and we're not going to go into all that regulation in this course, but there are transcription factors involved in both eukaryotes and prokaryotes. So the TATA box is the abbreviation, what we call the sequence in the beginning of the promoter is a bunch of A's and T's, and they just call it the TATA box for short. So that's part of the promoter. That's where transcription factors combine to enhance the binding of the RNA polymerase to the promoter. So there's a TATA box. And so there will be transcription factors which bind to the TATA box that enhances the binding of RNA polymerase to the promoter so that transcription can occur. So once again, there is a lot of regulation involved in transcription. We're not going to go into any detail about that regulation. So as the RNA polymerase moves along the DNA, it untwists the helix, so it opens it up about 10 to 20 bases at a time so that it can add nucleotides to create the messenger RNA. And just remember that it adds a U instead of a T. So RNA has no thymine in it. It's all uracil. So as the polymerase moves along, it's opening up the DNA, it's transcribing, and the DNA closes behind it. So a single gene can be transcribed simultaneously by more than one RNA polymerase enzyme. So one RNA polymerase can hop onto the promoter and start transcribing. Another RNA polymerase can hop on right behind it and start transcribing. And another one can hop on right behind that one and start transcribing. So multiple RNA polymerases can be transcribing the same gene simultaneously, one after the other, so that a lot of messenger RNA transcripts are being made. So it's just a way to ultimately make a lot of proteins, a lot of polypeptides, a lot of enzymes from that one gene, because many messenger RNA transcripts are being produced. As I mentioned before, messenger RNA is very short-lived inside of a cell. So if the cell wants to keep on making those proteins, it has to keep on transcribing and making those messenger RNAs. And that's a mechanism of regulation, right? A way for the cell to regulate the amount of those proteins, polypeptides, enzymes, whatever that they're making. Some cells make a lot of proteins. So if you take a cell like a B lymphocyte, and a B lymphocyte is a very important immune cell. It's the one that ultimately synthesizes antibodies. And antibodies are special immune proteins. And when that B cell is stimulated to make those antibodies, the entire internal metabolism shifts to protein synthesis, and it's just making tons and tons of those antibodies to secrete them. So you would have a lot of RNA polymerases hopping onto those genes to make that antibody. So there'd be a lot of messenger RNA transcripts being made, a lot of translation to make those antibodies, and then they're secreted because all those antibodies, they have to go all throughout your entire body, right, to encounter that pathogen wherever it may be. And so huge amounts of protein synthesis are occurring in an activated B cell, which is making those antibodies. So the RNA polymerase continues to transcribe until it reaches that terminator sequence and then falls off. So in prokaryotes, the RNA polymerase falls off immediately. And so in prokaryotic genes, there's no extra nucleotides, no extra stuff there, because the prokaryotes have to be efficient. They carry the genes that they need in order to survive. And remember, they have a circular chromosome. When it comes to eukaryotes, the RNA polymerase just kind of continues on past the terminator sequence for a while, maybe for hundreds of nucleotides past the terminator sequence. So eukaryotes, I don't want to say they're sloppy when it comes to transcription, but they will be modifying their messenger RNA, which prokaryotes do not do. So there's no extra stuff in the prokaryotic messenger RNAs, but eukaryotes have extra stuff, which gets removed. So let's look at some of those modifications that occur. So, and this is just in eukaryotes, not prokaryotes. So at the 5' end of the pre-messenger RNA, so the initial RNA transcript, there is a guanine, which is added to the 5' end. And we call that a 5' cap. So a guanine nucleotide is added to the 5' end. So that helps to protect the messenger RNA from hydrolytic enzymes because, again, messenger RNAs are short-lived molecules. And so a 5' cap helps to stabilize them. They have to be transported from nucleus to cytoplasm, so they got to last long enough, right, to reach a ribosome so they can be translated. And the 5' cap also functions as an attach here signal for the ribosome. On the 3' ends, a bunch of adenines are added, and we call this a poly A tail. So anywhere from 50 to 250 adenine nucleotides are added to the end of that RNA transcript. And so both of these modifications help to stabilize the messenger RNA molecule, help to be transported to the cytoplasm where it can hook up with a ribosome. This does not happen in prokaryotes. There is no 5' cap, no poly A tail, right? Because in prokaryotes, transcription and translation can occur simultaneously. So no modifications to the messenger RNA in a prokaryote are required. All this happens in eukaryotic cells. Now, it's interesting to note that certain viruses, which are RNA viruses, so their genomes are made of RNA. If they are eukaryotic RNA viruses, their genome looks like what you see here. There's a 5' cap and a poly A tail right to that viral RNA genome. So when RNA viruses are replicating inside of a host cell, they actually first have to convert their RNA back into DNA using reverse transcriptase. And then the host cells can transcribe that DNA into RNA, which is both a messenger RNA to be translated to make viral proteins. It's also at the genome of that virus. So an RNA virus is basically a messenger RNA transcript. It's carrying its genome made from a eukaryotic cell. So it's got a 5' cap and a poly A tail. Not all of them, but many of them do. So the most remarkable RNA processing is called RNA splicing. Again, this only happens in eukaryotes because eukaryotic genes have these intervening sequences called introns. So introns basically are non-coding regions. So think about this now. You have a gene, it's got all those nucleotides, and they're going to be read in groups of 3 in order to determine the amino acid sequence of a polypeptide or protein. In the middle of that are these nucleotide sequences which are not part of that protein, but they're there in eukaryotic genes. So, doesn't that sound kind of bizarre? Why would you have these long stretches of non-coding nucleotide sequences in the middle of your gene? If they don't code for amino acids found in the final product, why are they there? And eukaryotes have these, not prokaryotes. I mean, doesn't that sound strange, right, to have these introns in your genes? Sounds strange to me. Sounds strange to you guys? Hello out there. Support purposes, such as what, Ashari? What kind of support? So,
Could it be designed to give the cell a break or something while it's doing all the processing, just like a computer
break,
program?
it makes a lot of extra work for the cell because of the messenger RNA transcript. The introns have to be spliced out. They have to be removed in order to get your final functional messenger RNA transcript. Because if any part of the intron is left in, it's going to code for extra amino acids, which is going to mess up your protein. So this, to me, it's like, well, now the cell has to do this extra work to remove this extra DNA, which doesn't serve a purpose with the extra RNA, because the DNA is still there. But the extra RNA that's in the RNA transcript, the cell has to remove that, and it's got to remove it precisely, exactly correctly. Otherwise, it's going to screw up the final protein. I mean, that sounds pretty dangerous to me and pretty unnecessary. But it exists. Almost all eukaryotic genes have these introns. Not prokaryotic genes, but eukaryotic genes. So all of the good coding stuff are called exons, and the non-coding stuff is called introns. But the introns must be removed from the messenger RNA in order to form the final functional messenger RNA transcript. If that does not happen, the final protein product is not going to be functional. It's not going to fold correctly, right? Because it's got extra amino acids there. So the introns do have to be spliced out and spliced out correctly. So eukaryotes have to do this. So almost all eukaryotic genes contain introns. Introns are not part of the final protein, right? All the amino acids that might be encoded by introns are not there. in the end. So why are they there in the beginning? Like I said, it's an awful lot of work for a eukaryotic cell to do, and if it doesn't do it exactly correctly, that final protein's screwed up. Well, I will not keep you in suspense, but we will get to this later. But as it turns out, eukaryotes have the ability to splice the introns out differently. And so they call this alternative RNA splicing. So what that means is you can have one gene, just one gene, which can code for more than one protein product because the introns get spliced out differently. And that results in different messenger RNA transcripts and different protein products. So one gene can code for more than one protein product as a result of alternative RNA splicing. Prokaryotes cannot do this. Prokaryotes do not have the machinery, which is called the spliceosome, in order to splice out those introns. So then now a question should be occurring to you. If prokaryotes do not have introns and they cannot remove introns, and I just told you that we clone the gene for human insulin in bacteria, do you see a problem? Do you see a problem? So let's say this is the gene for human insulin, or it's the messenger RNA that's made from the gene for human insulin. It's got introns. So if you took the actual gene for human insulin, clone that into bacteria, they are going to produce this messenger RNA, which contains introns. And they're going to translate that to make a protein, which includes the introns. That protein's not going to be functional because it's got extra amino acids that don't belong there. So, but they did clone the gene for human insulin into bacteria. How did they do that? Given this problem of introns, can you think of a way to solve that problem? I'll wait. Well, how would you do that, Laura? They removed the introns before inserting it. Did they use little microscopic scissors for that, Chris? So how would you actually go about doing that? Right, the problem is the eukaryotic gene has introns. We want to stick that gene into bacteria, but if we do that, the introns are going to be transcribed and translated to make a non-functional protein. So that's no good. How can we solve that problem?
Put the spliceosome in the bacteria.
What about the bacteria, Zoe?
Put the spliceosome in the bacteria.
Oh, the spliceosome is a complex thing to create, and bacteria don't have that, so that would create a lot more potential problems. If you tried to clone a spliceosome into the bacteria and do it that way, I don't think that works because there's a bunch of different proteins and some small nuclear ribonuclear proteins that come together to help make this thing. So that doesn't really work to do that. A nice idea, but that gets even more complicated. So what could you do? Well, we know that eukaryotic cells do make something that's nice and good, right, with all the introns removed. So you could isolate the processed messenger RNA where the introns have been removed already, right? So you take that processed messenger RNA with all the introns removed, right? So you obtain that. Well, now you've got an RNA with no introns. What can you do with that?
Reverse it to a DNA.
Exactly, right? So now we can use reverse transcriptase, right, to make, oops, sorry about that, to make DNA from RNA. So now we have what I call a clean gene, right? No introns. Okay, so what do you do with that? You can stick that clean gene, whoops. We can use the process of transformation to insert that gene into bacterial cells. Next week in lab, we're going to talk about a transformation and see how that can be done. So, right, because we learned about the enzyme reverse transcriptase, now we have a way to clone any eukaryotic gene into a prokaryotic cell. We isolate the processed messenger RNA where all the introns have already been removed by the eukaryotic cell. We then use the enzyme reverse transcriptase to make a DNA copy from that processed messenger RNA. Now we have a clean gene with no introns. And it's relatively easy to insert that gene into bacteria using the process of transformation. Sound good? Yes, we're good. As soon as I change slides, this all disappears. So speak now. So the spliceosome will remove the intron from the messenger RNA transcript in order to produce the final functional messenger RNA, which is all exon. It's all coding information. But as I said, sometimes the cell can splice out the introns differently and produce different messenger RNA transcripts which code for different proteins. So eukaryotes have a way to add some more variation to their genome without adding more genes. So one gene could potentially code for multiple, protein or polypeptide products. Prokaryotes can't do this, right? So they got to have a specific gene, for every protein product they're making. But eukaryotes, maybe just have one gene and produce 3 different protein products from that same gene. So instead of needing 3 genes, they just have the one. So those small nuclear RNA proteins act as a ribozyme. So a ribozyme is an RNA molecule that functions as an enzyme. So here we see an example of an enzyme that's not made of protein. It's made of RNA. So this was a huge discovery. I think it was back in the 70s, maybe early 80s. This was huge. Right? The idea that RNAs can be enzymatic. Right? Because before that, we thought only proteins were enzymes. But now we know that RNAs can be enzymatic. So this was huge news when they discovered it way back when. So there are other ways to splice out transcripts besides spliceosomes. But for eukaryotes, we focus on the spliceosomes. And then prokaryotes don't have spliceosomes. So all biological catalysts are not in fact proteins, right? Because we now know that RNAs, can also be catalysts. So, and so I mentioned before that the first molecule to use and store information was most likely RNA. Later that led to DNA. And so we know that RNAs can catalyze the duplication or replication of RNA molecules. And so now we have some evidence which shows RNAs can act as enzymes. RNAs can self-replicate. And so RNAs were probably the first type of nucleic acid molecules that evolved to carry information and form those primitive ribocells or ribocytes, which were based on RNA. Later, that led to the evolution of DNA. So, all some pretty interesting stuff now that we can put these things together and see how life may have evolved, starting with primitive RNA molecules. So, I'm going to stop here for today. We'll continue talking about the alternative RNA splicing tomorrow, and then finish up discussing translation, right? Actual protein synthesis. So any questions or comments?